Hi, my name is Sohail. Today I am going to discuss part 4 of eukaryotic transcription. Main points of the lesson are eukaryotic elongation, fact, facilitates chromatin transcription, eukaryotic termination and mRNA processing. During transcription initiation, DNA unwinds and unzips at the promoter, which is a specific start site for the beginning of each gene. Proteins called transcription factor bind to the promoter and also bind to the data box in the promoter region, and then transcription factor they recruit an enzyme which is called RNA polymerase II enzyme to form transcription initiation complex. During eukaryotic elongation, the polymerase enzyme it is released from the transcription factors, and transcription is allowed to proceed as it does in case of prokaryotes with polymerase synthesizing pre-messenger RNA in the direction of 5' to 3'. RNA polymerase II enzyme transcribes the major part of eukaryotic gene. The enzymatic process of elongation is essentially the same in eukaryotes and in prokaryotes, but the DNA template is more complex in eukaryotes. When eukaryotic cells are not dividing, their genes exist as diffuse mass of DNA and proteins that is called chromatin. The DNA is tightly packed around charged histone proteins at repeated intervals. Histones are basic proteins and their positive charges allow them to associate with DNA which is negatively charged. So, these DNA histone complexes collectively known as here nucleosomes, and nucleosomes are regularly spaced, and each nucleosome that has 146 nucleotides of DNA wound around 8 histones like thread around a spool. For polynucleotide synthesis, that is pre-messenger RNA, the transcription machinery needs to move histones out of the way every time it encounters a nucleosome. This is accomplished by a special protein complex that is called FACT, FACT, which stands for facilitate chromatin transcription. So, this complex pulls the histones away from the DNA template as the polymerase it moves along it. Once the pre-messenger RNA is synthesized, then the FACT complex replaces the histones to recreate the nucleosome. After eukaryotic elongation is the termination, and termination of transcription is different for different polymerases. Unlike in prokaryotes, elongation by RNA polymerase II in eukaryotes takes place 1000 to 2000 nucleotides beyond the end of the gene. So, this pre-messenger RNA is removed by cleavage during mRNA processing. So, during elongation, RNA polymerase II enzyme that synthesizes pre-messenger RNA in the direction of 5' to 3'. And RNA polymerase II transcription, termination and 3' end processing are two different processes, but termination and 3' end processing they are closely connected and influence each other. So, this eukaryotic pre-messenger RNA undergoes extensive processing before it is ready to be translated. So, once the elongation is completed, it proceeds through RNA processing events, that is polyadenylation and termination. Polyadenylation that occurs at 3' end of messenger RNA, which is linked with the termination of transcription. Eukaryotic mRNA operate for several hours, whereas typical E. coli mRNA remain usable no more than 5 seconds. So, this pre-mRNA is first coated with RNA stabilizing protein, and that protein protect the pre-mRNA from degradation while it is processed and exported out of the nucleus. So, three most important steps of pre-messenger RNA processing are 5' capping, 3' polyadenine tail and pre-mRNA splicing. What is 5' capping? While the pre-mRNA is being synthesized, a 7-methyl guanosine cap is added to 5' end of pre-messenger RNA by a phosphate linkage, and this functional group protects the mRNA from degradation. In addition, the factors involved in protein synthesis, they can recognize the cap to help initiate translation by ribosomes. What is 3' polyadenine tail? Once the elongation is complete, the pre-messenger RNA is cleaved by an enzyme that is called endonuclease between adenine uracil adenine consensus sequence and a guanine uracil rich sequence, leaving the adenine uracil adenine sequence with pre-messenger RNA. So, an enzyme which is called polyadenine polymerase enzyme that adds a string of approximately 200 adenine nucleotides, which is called polyadenine tail, and this modification further protects the pre-messenger RNA from degradation, and it is a signal the export of cellular factors that transcript needs to the cytoplasm. What is mRNA splicing? Eukaryotic genes are composed of exons which correspond to protein coding sequence. So, exons are expressed, and intervening sequence which is called intron. Introns, they have intervening role which may be involved in gene regulation, but they are removed from pre-messenger RNA during processing. So, intron sequences in mRNA do not encode functional protein, and exon sequence that encodes for functional protein. The genes of higher eukaryotes, they contain one or more introns, and these regions may correspond to regulatory sequence, but the biological significance of having many introns or having very long introns in a gene is unclear. It is possible that introns slow down the gene expression because it takes longer to transcribe pre-messenger RNA with lots of introns. All of pre-messenger RNA introns must be completely and precisely removed before protein synthesis. If the process make a mistake by even a single nucleotide, the reading frame by rejoined exons would shift and the resulting protein would be dysfunctional. So, the process of removing introns and reconnecting exons is called splicing. The splicing of pre-messenger RNA is conducted by complexes of proteins and RNA molecules called spliceosomes. On the other hand, RNA polymerase 1 and 3, they require termination signals. The gene transcribed by RNA polymerase 1 contain a specific 18 nucleotide sequence that is recognized by a termination protein. So, RNA polymerase 1 requires a protein factor to terminate the synthesis of RNA. The process of termination in RNA polymerase 3 involves a mRNA hairpin similar to row independent termination of transcription in prokaryotes. To sum up, transcription initiation complex that consists of transcription factors and RNA polymerase 2. During elongation, RNA polymerase 2 is released and mRNA molecule is synthesized in the direction of 5' to 3' from DNA template strand. The DNA strand that is transcribed, that is called template strand, and the other one is called non-template strand or coding strand. The termination of transcription in RNA polymerase 2 synthesizes 1000 to 2000 nucleotides beyond the end of gene, and this pre-mRNA tail is removed by cleavage by endonuclease enzyme between uracil adenine consensus sequence and guanine uracil rich sequence. An enzyme called polyadenine polymerase that adds a string of around 200 adenine nucleotides to 3' end to form polyadenine tail. 5' capping is 7-methyl-guanosine cap is added to 5' end of growing mRNA molecule by a phosphate linkage. And pre-mRNA splicing is a process of removing introns and reconnecting exons. The removed introns are degraded.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.